Hey, welcome to Let's Get to the Marks, going through AS Biology Paper 2. This is the 2022 June paper. There are 9 questiones. It's the paper 2 for the AS Biology AQA syllabus. And why don't you pause the video at the start of each question, have a go, then unpause the video and watch the walkthrough. So the first two parts are 1.1, what is a gene? So it's a one marker, but essentially you need to know the definition of a gene off by heart. 1.2, describe how the production of messenger RNA, mRNA, in a eukaryotic cell is different from the production of mRNA in a prokaryotic cell. Why don't you pause the video, have a go, and then unpause and watch the explanation? Okay, so what is a gene? Well, let's go through. So at GCSE, our definition of a gene is just really a short section of DNA, but we need to be a little bit more specific at A level. And we need to actually say a little bit more specifically what the gene is. So, what is this when we look more closely at it? It is a specific sequence of DNA nucleotides that code for a specific protein. So we need to be a little bit more accurate at A level, and we want to say a specific sequence. Oh my gosh, it would help if I could spell the word sequence. A specific sequence of DNA nucleotides, and we want to say the code for a specific protein. You know, it's that specific DNA sequence that code for the different amino acids that make it. I could probably make a little bit more detail and say a specific sequence of DNA nucleotides that code for an order of amino acids to make a specific protein. Let's just add in the order of amino acids that code for a specific uh, order of amino acids. Uh, that code. Oh my gosh. Talk about make the definition super long. But I think the keys here are that you mentioned the word DNA nucleotides. Um, yeah, and that they code for protein. Okay, 1.2, describe how the production of messenger RNA in a eukaryotic cell is different from the production of mRNA in a prokaryotic cell. So in a eukaryotic cell, it happens in the nucleus, doesn't it? Whereas a prokaryotic cell doesn't have a nucleus. So in eukaryotic cell, it occurs in the nucleus. In prokaryotes, of course, they don't have a nucleus. So in prokaryotes, it does not happen in the nucleus. Now, I don't know why I'm overwriting this answer because you actually would just need to say in eukaryotes, it occurs in the nucleus, or in prokaryotes, it doesn't happen in the nucleus. Also, prokaryotes you might have heard of splicing. Also, prokaryotes do not need to remove introns, so there is no splicing. Now, technically, there are a certain type of prokaryote called RK, which you've heard with the three-domain system made by Carl Woese. There are actually RK prokaryotes that do still have some introns. So to say all prokaryotes don't have introns is technically incorrect, but at A level, they'll allow it for you to say the prokaryotes don't have introns, and so they don't need splicing. What does this mean? Well, actually, in prokaryotes, they have a little bit more advanced than A level, but they have something called coupling, which is where translation and transcription happen at the same time. So as their circular DNA is being um, transcripted into mRNA, at the very same time, the mRNA gets read by ribosomes and is then being translated into that specific sequence of amino acids. So what is that implications for the bacteria? Well, it means that they can make proteins much, much faster than eukaryotic cells. And therefore, one of the reasons why they reproduce so quickly is because of their rapid production of proteins, which is because of this coupling system. And actually, at some point in their evolutionary history, they've got rid of their introns. So it may be that bacteria in their evolutionary ancestors actually had introns, but in order to speed up their reproduction so that they could compete better, they got rid of introns to make reproduction and production of proteins faster. Okay, coolio. Enough whiffle, waffle, waffle. Let's move on. Okay. 1.3, scientists produced short single-stranded artificial nucleic acid called PNA. The PNA binds to a small section of DNA. So it's basically like a little bit of mRNA that's been artificially made in the lab. The scientists produced PNA into cells and discovered that these cells produce less mRNA than cells that did not contain PNA. So when they stick this artificial single-stranded DNA into cells, they find that those cells that have it don't make as much mRNA. Suggests how the PNA affected transcription of the section of DNA. Why don't you press pause on the video? This is an application question. What's happened here? I've got my crusty little diagram of PNA attached to our DNA. Guess what? Here is my RNA polymerase. So this is my RNA polymerase. What can't it do? Maybe the RNA polymerase isn't complementary to PNA, only to when the DNA is attached to mRNA. Whatever happens, maybe the all right, can't speak. Maybe the RNA polymerase cannot bind, but also we want our RNA-free nucleotides, so uracil, adenine, guanine, thymine, we want those to attach. If this PNA is in the way, these free RNA nucleotides can't attach. They're like, hold on a minute, mate. We want a complementary base pair to the template strand, but we can't. This blooming PNA is in the way. So we need to just put that down as an answer. So we're going to say PNA binds by comp base pairing. Now, don't write comp. You need to write complementary, but I can't bother to write it out. So PNA binds by comp base pairing to the DNA. And we could either go down the route of preventing RNA polymerase from attaching to the binding site, or we could say preventing RNA free nucleotides from binding to the template strand. So essentially, they're testing us on using the term complementary base pairing at some point, and the fact that the PNA is inhibiting these free RNA nucleotides from attaching by complementary base pairing to the template strand. So that's basically what they're testing us on, and there we go. Okay, 1.4, describe the role of ATP in the process of translation in protein synthesis. Okay, so I'm going to go through the explanation, describe the role of ATP in the process of translation in protein synthesis, 2 marks. So look, we've got our tRNA bringing amino acids. We've got the anticodons going to bind to the codon on the mRNA that's attached to a ribosome. And that bit might not require energy, but certainly in order to form the peptide bonds in between the individual amino acids, we're going to require some energy. And that energy is going to come from breaking down ATP to ADP plus inorganic phosphate to supply the energy to form the peptide bonds. Also, it may potentially be that the tRNA, as it picks up an amino acid, maybe as the amino acid binds to the unique tRNA binding site, remember there's 20 different amino acids, and we've got these binding sites on the tRNA, it may be that that requires energy as well in the form of ATP being broken down to ADP plus PI to release a little bit of energy. Let's put that into a 2 marker. So energy is required from the hydrolysis of ATP to ADP plus PI for the formation of peptide bonds. We want to say between amino acids, which is going to make of course, our primary structure, and for the binding of tRNA to the amino acid. I should really say the binding of the amino acid to this. It's really the amino acid that binds to the tRNA, but you get the point, peeps. What term, 2.1, what term is used to describe the different structures of alpha glucose and beta glucose? And it's worth 1 mark. Now, if you're a chemist, this is nice and simple. And hopefully, you know, they are isomers. So it's just isomers, guys. You could say isomerism, which is the name of the situation, or you could call them isomers. So they have the exact same molecular formula, C6H12O6, so the same amount of each element, just arranged in a different spatial arrangement, which happens to make them mirror images. You don't need to know that for bio. Yeah. Right. A student investigated the difference in the reducing sugar content of two fruit juices. He performed the biochemical test on each fruit juice using Benedict solution. He then used the colorimeter with each test result. Describe how the results from the colorimeter can identify the fruit juice containing the higher sugar content. So you can either measure transmission or absorption. So guys, so a higher level of absorption, higher absorption means a higher glucose concentration. Oh my gosh, higher glucose concentration, right? But you might not be measuring I swear I should put a P in absorption. Anyway, never mind. Right. Higher absorption means higher glucose concentration or lower transmission. So you might be measuring transmission. You might have a detector detecting how much light gets through and doing it as a percentage of the light that gets through. So you might say lower transmission means higher glucose concentration. So it all depends whether you're doing transmission or absorption and which one you prefer to do. So either the amount of light that gets absorbed is equal or correlates to the glucose concentration, or the amount of light that gets through. You're looking at how much light gets through rather than how much is absorbed. So you can do either one. The student controlled variables in the test tube using Benedict solution. Give two variables that are controlled. So if we're trying to measure the sugar concentration of these different solutions, that's what's going to be different. They looked at the two fruit juices. That's like their independent variable. What do they need to add about the fruit juices each time to make the test fair between the two fruit juices? So we want to say same volume of fruit juice and maybe same concentration of fruit juice, but definitely same volume of fruit juice, or maybe not same concentration, but we'll go with same volume of fruit juice, same number of drops of Benedict's. I don't know if that's even going to affect it. Same number of drops of Benedict's solution. What about the same temperature? When you do Benedict's, you have to heat it as well. So let's say warmed to the same temperature. I don't think that would affect the final mass. It would just affect how quickly you form the precipitate. So maybe that one's not so good. Same Benedict's is probably important. Same volume of fruit juice. I wonder if we could say same concentration of fruit juice as well. Why not? Because the fruit juice, you're not saying it has to have the same sugar concentration, but you're saying the same concentration of fruit juice in the solution. So let's go with same concentration of fruit juice. We're not saying the same sugar concentration. That would defeat the purpose because that's what we're trying to find out, what the sugar concentration is. But the same concentration of juice per volume of the solution. Okay, which is the same concentration. I think I've whiffle waffled enough. Apples consist of Apples consist of flesh tissue which surrounds the core tissue where the seeds are located. Yeah, we've all eaten a nice apple and then you bite into the core and it's all tough and nasty. Apple seeds have cyanide in them as well. So if you ate like about 100 apple cores, you could kill yourself. A student has an apple with cyanide, by the way, is a respiratory inhibitor for respiration enzymes. A student has an apple with a mass of 180 grams. The ratio of flesh tissue to core tissue in the apple is 5:1. So we've got a mass of 180 grams with a ratio of 5:1 between the flesh and the core. 8% of the whole apple is sugar. Calculate the mass of sugar in the flesh tissue. Ooh. So we want to find out, first of all, what is the mass of the flesh? And then we can work out how much the mass of the flesh is sugar. Anywho, peeps, what I want you to do is grab your calc, grab a pen, grab some paper, pause the video, have a go. Let's go. So first up, we've got our lovely apple. Now, our lovely apple has like a core, pom pom pom, and it has little seeds in there. Lovely jubbly. So we want to work out, this is a 5:1 ratio. Let's work out if the whole thing is 180 grams, how much flesh we have. And of course, this here is the flesh. So it's 5:1. So what I want to really do is divide this by 6. So I'm going to go 180 divided by 6 equals 30 grams. And essentially, that means that I'm going to be in a ratio of 150 grams. So one is 30 grams, and then I'm going to be 150 grams of flesh, and there's going to be 30 grams of core. So that's the flesh. The mass of the flesh is 150. The mass of the core is 30, and that gives me a 5-to-1 ratio. Now, so we now know the mass of the core, and we know that 8% of the whole apple is sugar, which means that 8% of the flesh is sugar. So I want to do 8% of this. So what are we going to multiply it by? Well, we're going to multiply it by 0.08. Or you could say multiply it by 8 over 100, times it by 8 over 100, or just times it by 0.08, and that is going to give us the mass of sugar in the flesh. So 150 times the 0.08, that equals 12 grams of sugar in the flesh of the apple. Lovely jubbly. Iodine solution stains fresh apple tissue black. When iodine solution is added to apple stored for a week, the stain is less black. The water potential of the apple juice decreases when the apples are stored. So just why the water potential of the apple juice decreases when the apples are stored. So you don't really need all of this. We can just say why the water potential of the apple juice decreases when the apples are stored. But this is to give you a clue. So I'll explain why they gave you this little backstory and how this was supposed to help you come up with your answer. You can just come up with the answer from this, but you this can give you a little clue. So why don't you pause the video, have a go, and then I'll go through the explanation. All right, let me go through the explanation. Guys, what we're going to go through here is, right, the iodine was black, right? The apple went black, meaning there's lots of starch. And then 1 week later, there's less starch. Why is that? What has the starch been converted to? So starch has been hydrolyzed, which means broken down, into glucose, alpha glucose, right? The glucose is soluble, whereas starch is not soluble. The glucose is soluble, so lowers the water potential of the cells, right? So what that's going to mean is it's going to have an osmotic effect and cause water to move in the cells and the cells to swell up. They could have then followed this up with why did the cells become more turgid? And that's because water moves from a high water potential outside the cells into the cells because now they have a lower water potential because of the glucose. So, but you get the idea. Having soluble solutes like glucose or salt inside your cell lowers your water potential. There we go. And that's because the starch got converted into glucose. Right, let's move on. Okay, so question 3. Figure 1 is the image of part of a cell lining the ileum. So the ileum, you might be asking yourself, where the blooming is the ileum? The ileum is in the small intestines. So hopefully you know that. Oh my gosh, small intestines. Now what you need to do, small intestines. Now what we need to do, oh, we've got microvilli labeled there. Describe and explain 2 ways the structure shown in figure 1 increase the rate of absorption. Oh, look, the absorption has a P. I love spelling it with a P. Absorption by this cell. So we need to say 2 ways that structure is shown increase the rate of absorption in this cell. So first of all, what is it that microvilli do? And this is a single cell. These are not cells. This is a villus. So this gives the small intestine a large surface area by its nature, having lots of villi in your small intestine. But these are microvilli. These give the individual cells, the villus, a large surface area. So what we want to say is the microvilli give the cell a large surface area. Now, it does say and explain, which increases the rate of absorption. Okay, so the large surface area increases the rate of absorption. This is going to get us a mark. Now, what else? What do we think about the cell membrane here, or the thickness of the membrane here around the microvilli? I'm going to go with the fact that it's an exchange surface and the thickness of the membrane must be quite slim. Those fatty acid tails in the phospholipid bilayer must be quite short in length, giving the cell membrane a thinness. So I'm going to say thin membrane creates, oh my good gosh, xararites, right, creates a short diffusion pathway. Um, there we go. Thin cell membrane. So don't say thin cell wall because, of course, an ileum is not found in plants, it's in animals. And so they don't have cell walls. So there is a thin wall to the small intestine, but we are looking individually at this cell, not at the tissue. So this villus creates part of the wall of the small intestine, which is a tissue, but the cell itself is not a wall, so we can't say has a thin wall. It has a thin cell membrane. Got to be very careful there. And then I'm guessing, are these mitochondria or are these look these could be mitochondria. These are mitochondria here, these sausage-like shapes. So let's go with lots of mitochondria, right? Lots of mitochondria, lovely jubbly, means lots of ATP for active transport. There's another answer we could go with. That one's quite nice as well. And there's my three answers. Um, oh my God, transpoport. Yes. Don't check that for spelling, guys. Boom! Oh no, more maths. An ileum cell without microvilli has a 7.85 micrometer square of cell surface in contact with the digested substances, amino acids, glucose, glycerol. You get the idea, fatty acids. A scientist found an ileum cell with 1,000 microvilli. Lovely. The microvilli cover the entire cell surface in contact with the digested substances. Okay, microvilli are zero point 0.1 micrometer in diameter and 1 micrometer in length. The surface area of the microvillus is calculated using this equation. 2 pi RL, L being length, plus pi R squared, where pi is 3.14, R is the radius, L is the length. Calculate the ratio of the area of the ileum cell surface with microvilli to the area of the ileum cell surface without microvilli. Show you're working. It's worth 2 marks. This is a beep of a maths question with loads of parts to the stem. Why don't you pause the video, have a go, see if you can get both marks, and then unpause, and I'll go through the explanation. Let's look at what we got here, peeps. So I know there's 1,000 microvilli. I know the microvilli are zero point 0.1 in diameter and 1 micrometer in length. But look, I'm using radius, not D for diameter. So I want to do no. Oh, let's change to black, please, Bob. And we're going to go zero point 0.1 divided by 2. So my R is 0.05, and that is R. So that is my R is 0.05. Okay, got the radius. Let's go. Let's work out. Oh, if I was to set this out as a ratio, well, I know that the without microvilli, oh my gosh, this is so sloppy. That looks like a 3. Oh my gosh, and what the hell is that? Right. That is 7.85 micrometer squared. And this is going to be without. And then over here with. So we're doing with microvilli to without, and we need to work out what that is, then simplify and get our ratio. Okay, let's do it, peeps. So it's just really a case of putting this in. So it's 2 times 3.14 times the radius, which is 0.05, not 0.01, so 0.05 times the length. What was the length? The length was 1 micrometer. And then we're going to add this to 3.14 times the radius 0.05 squared. Now, this is a pain in the butt. You can jam this all in your scientific calculator if you know what you're doing. If not, do that. 2 times 3.14 makes 6.28 times by 0.05 separately, and then times 1. And I think you're going to get something like 0.3 14 for that bit. And then we're going to add that to 3.14. So again, if you're really rubbish with the calculator, just do 0.05 squared. You're going to get like 0.0025, whatever. And then times that by 3.14 separately, if that's what you want to do. And then if you do that, you're going to get 0.0785. And then what you're going to do, you're going to add those together. And if you add those bad boys together, you're going to get something like, so if we add these bad boys together, now you can just jam this all in your calculator. You don't need to do this long ass way like this. Then you're going to get 0.32185. We go, well, look, that's definitely smaller. What's the point in having microvilli if your surface area is going to be smaller than without? And then we realize, oh, because of course, there are 1,000 microvilli in one cell. So we need to times by 1,000. And once we times by 1,000, we're going to get 321.85. Now you can round, obviously, but I'm just going to keep my numbers like that. So that is the surface area with, and that's without. What I need to do now is divide both by the smallest. That's how we're going to get 1 over here, because we're going to divide that by 785, and then we're going to divide this by 7.85. And if you do that, you're going to get 41 to 1. But probably if you round on the way, you might get a slightly different answer, but there's probably a range between 40 and 41. So there you go. 41 to 1 is the answer. Boom, 2 marks. Lovely. Don't forget to like, share, subscribe, all of that business. All right, I am moving on to 3.3. The protein ZO1 is found on the surface of ileum cells. The scientists used an anti-ZO1 monoclonal antibody to identify ileum cells in a sample of intestine observed using an optical microscope. That's just a light microscope. So basically, they've looked at a sample of ileum cells and they basically observed this protein ZO1 by having an anti-ZO1 monoclonal antibody. Suggest how the monoclonal antibody helped the scientists to identify the ileum cells in the sample of the intestine. Why don't you grab your pen, pause the video, and have a go? All right, so what we want to explain here, guys, is that essentially these ileum cells, unlike other cells, have this unique ZO1 protein, kind of like an antigen, on their cell membrane. And if we make a ZO1 antibody monoclonal, so thousands of identical antibodies, you're going to get antigen antibody complex formed with the monoclonal antibody and that protein. What that's going to mean is that you can then look for the cells that have the antigen antibody complex, and then you will have identified the ileum cells. Now, how can you identify that? Maybe you have an enzyme and a substrate. Maybe you have all sorts of different marker. You have a marker on that antibody that allows you to identify it. So yeah. Wunderbar. So you could have a fluorescent marker, a dye. You could try something with an enzyme attached to the monoclonal antibody, and then you add a substrate. And if the enzyme if the antibody is present with the enzyme attached, then it will catalyze the substrate, which involves a color change. So that could be something. So some sort of marking system so you can identify ones where the antibody antigen complex kind of has formed, or the antibody and protein complex has formed. So what are you going to get the marks for here? Because there's 3 of them. Well, you're really going to get the mark to say that the antibody binds to the protein, specifically only to that protein on the cell surface membrane of the ileum. You know what? Just because they are really, really fussy, I've said the cell surface. What I need to add is the word membrane, just in case, because you never know. You'd hate to miss it out because you said cell surface and not the membrane. Forming the antigen and blah, blah, blah. We can identify them because they've been marked, and that is really your 3rd mark. Lovely jubbly. That's the end of question 3. Question 4, describe how the process of meiosis results in haploid cells. Do not include descriptions of how genetic variation is produced in meiosis. Okay, and it's just describe how the process of meiosis results in haploid cells, 4 marks. So we only need to describe. They're not interested in how we get genetic variation, and there are 3 factors that get that. So it's really just a case of going through meiosis. So you might be thinking to yourself, do I need to talk about prophase, telophase, all these different things, basically PMAT. And of course, with meiosis, there are 2 cell divisions. So you're going to have PMAT twice, prophase, metaphase, anaphase, telophase twice. So you're like, oh my God, do I have to reel off all of that? Some details might get you a mark just from the examiner being generous, but this is just an overall description of the process and how we get haploid cells. So what specific parts of those result in the separation of the chromosomes so that we end up with haploid cells N instead of 2N. So what we want to say, first of all, is DNA is replicated in the interphase, actually in the S phase, isn't it, of the interphase. So it's in the synthesis phase of the interphase. The DNA is replicated. Then we want to say during meiosis one, which phase is it where the homologous pairs of chromosomes separate? And so that is going to be during the anaphase, the homologous pairs separate to yeah, different ends of cell. Yeah, so what's the difference? Well, homologous pairs are these bad boys. So they are being separated so that you end up with, in the first division, they're like that. But this is still a diploid cell, okay? So this is still going to be diploid. And then what we want to say is during anaphase 2, it's kind of during late prophase 2, early anaphase, they get separated, don't they? So during anaphase 2 of meiosis 2, what do we call these bad boys that are joined together by a centromere? We call them sister, because they're identical, chromatids, even though sisters aren't identical. So it's kind of weird. Maybe they should be called twin chromatids. That would be a more intelligent name, but never mind. Right. During anaphase 2 of meiosis 2, the sister chromatids are separated. Obviously, the centromeres pull apart when the spindle fibers shorten or contract, and they rip apart the two chromatids to each pole. Okay, and then what you're going to essentially do this time is, of course, I have only used one chromosome. There'd be 46 of these bad boys. And so there you go. You have a haploid daughter cells forming haploid daughter cells. Forming haploid daughter cells. So that's oh my gosh, it didn't come out. Where's my text box? I don't know what that's doing. Forming haploid daughter cells. And here we go. So really what our marks are for is explaining that in order for you to get haploid cells, you have to double the DNA first. If you don't double the DNA first and talk about the interphase first, then really you're explaining that they've divided and divided again. You're not going to get haploid like that with two divisions. Without a doubling, you're going to end up with a quarter, not half the chromosomes. So we don't want that. We have to double first, then half, and you get a quarter. Okay. So during meiosis 1, we're going to divide the first time, and this time it is the homologous pairs of chromosomes that separate. And then during anaphase 2 of meiosis 2, the sister chromatids are separated, and we get 4 haploid cells. Right, let's go. 4.2, figure 2 shows the arrangement of chromosomes in a cell during the first meiotic division. Lovely. Okay, so this is during the first meiotic division. I guess we are on the spindle fibers. We're all connected on the spindle fibers, late prophase, and we're metaphase, sorry, late metaphase. I don't want to talk about prophase. Late metaphase, we're all connected to the spindle fibers and we're going into the anaphase. The scientists observed 300 cells. All of the 300 were at exactly the same stage of meiosis as the cells shown in figure 2. Right, so all 300 cells were at this stage. Use your knowledge of independent segregation of homologous chromosomes to calculate how many of these cells are expected to have an identical arrangement of chromosomes to those shown in figure 2, assuming no crossing over occurs. So let's read through this again because I don't really understand the question. The scientists observed 300 cells. Get that. All of the cells were at the exact same stage of meiosis as shown. So they were all in the anaphase or metaphase. This looks like the metaphase. Use your knowledge of the independent segregation of homologous chromosomes to calculate how many of these cells of the 300 are expected to have an identical arrangement of chromosomes to those shown in figure 2, assuming without any crossing over. There's a little bit of a brute of a question, but it's basically saying how many different arrangements the homologous pairs have to stay like this, right? Because this is homologous because it's the same length. This is like mother's chromosome. This is dad's chromosome. This is like chromosome 1 and chromosome 1. Chromosome 1 from dad, chromosome 1 from mum. I can't start moving them about to other ones. They line up in homologous pairs. Look, These are identical in size. Their alleles will be different because they're from mum and dad, but they're identical. So how many different arrangements of these, keeping the fact that the homologous pairs have to be together, can we make? Well, I've got white, black, black, white, black, white, black, but I could have white, black, white, black, white, black, white, black, or I could have black, white, black, white, black, white, black, white, or I could have black, white, black, white, white, black, white, black. So how many different arrangements are there? There's four, and there are two arrangements in each of those. So actually, the number of arrangements is quite easy to calculate. It's 4 to the power of 2, and that equals 16. So 4 squared. There are 16 possible arrangements of the homologous chromosomes here. Of course, in a human, that would be 23 to the power of 2. So it'd be a lot more different possible arrangements. So we've got 16 possible arrangements. There are 300 cells. So how many would have this exact? Well, 1/16 of 300 would have that exact arrangement. So you could do 300 divided by 16, and you'll get something like 18.75 cells should be at any one time exactly the same as this arrangement. And so I don't know what that is. Is that 19? Do you round that to 19 or 18.7, or do you just say 18? I'm just going to go with 18, although I could round to 19. I think they'll accept either. Okay, I'm going to go 18. There we go. Bosh, all done. Draw a diagram to show the chromosomes in one gamete produced by meiosis from the cell shown in figure 2. So I need to do one gamete. So if we go back up here to do it and draw a gamete produced by meiosis from this, well, remember this is in the first, this is in this meiosis 1, but when we get to meiosis 2, the chromatids are going to separate. So I need one chromatid from each of these, and that is it. So my gametes, it could be a sperm with a little tail, or it might not be a sperm. It might be an egg, in which case it won't have a tail. Right, let's zing in. So it's going to have one, then one of the baby ones, beep boop, beep boop, then one of these ones, and then one of these ones. So essentially, the mark is going to be for you showing 4 chromatids. And you might say, why is it only 4? What does it have? Well, during meiosis 1, the homologous pairs separate. So if it was meiosis 1, the end of meiosis 1, you will have one of these homologous pairs in each cell. But then at the end of meiosis 2, the chromatids, sister chromatids separate. So you're going to just end up out of this, you get one sister chromatid. Because, of course, we're going to make 4 daughters. So each of those 4 daughter cells in meiosis has to have one of these chromatids in it. So that is our answer for 1 mark. 4 chromatids in a cell. Boom! Let's move on. Oh no! Not the spermatophite. Right, 4.4, figure 3 shows the life cycle of a fungus. Of course it does. The life cycle includes sexual reproduction. Oh, goody, exciting. Figure 3, diploid cells in a mature zygote. You can start anywhere, it's a cycle. So diploid cells in a mature zygote, spore producing stage. This is making spores. Then we have A, diploid spores are produced, and then they go off into these weird little things, look like hyphae or something, mycelium maybe, and then growth and development. What is the process shown by arrow A? Well, let me shrink it down so we can actually see. This is actually simpler than we expect. These are diploid cells and these are diploid spores. It doesn't tell us anything about the spores, so I'm going to assume that that has to be mitosis, because if it was meiosis, these would have to be haploid. So we are going to go with mitosis. Now, and my clue there is that it's diploid before and it's diploid after. So it has to be mitosis because you're doing identical, right? If it was fertilization, you'd be going from haploid to diploid. So it's not that. If it was meiosis, we'd be going diploid to haploid. It's not that. And if it was binary fission, we'd kind of be going diploid to I would be going diploid to diploid, but binary fission happens in bacteria, not fungi. So that's why we get rid of that. Okay, now let's zing on. Oh, that's the last part of the question. Boom. I've done a lot of booms. Maybe I should calm down with the booms. Okay, 5.1. Describe how organisms are grouped in phylogenetic classification system. Describe how organisms are grouped in a phylogenetic classification system. I learn how to speak properly for once. Phylogeny is how we organize them in this situation here. We organize them into a hierarchy: class, order, family, genus, species, right? So we order them kingdom, class, domain, kingdom, class, order, family, genus, species. And there is no overlap between the different groups. So grouped in a hierarchy um, with no overlap. So there's no overlap between those groups in the hierarchy according to their evolutionary similarities. So, including to their evolutionary, let's say, relationships. So, obviously, if you are in the same species, you are more similar than something that shares the same genus but is in a different species. So, as we go down the hierarchy, they become more closely related in evolutionary terms. But there's no overlap between the hierarchies. And this is just a revision card job. You need to know that off by heart. Okay, let's go. Table 1 shows some taxa in the phylogenetic classification of a rhinoceros species. Figure 4 shows the relationship between the taxa in the classification of two rhino species, the rhinoceros unicornis and Disorhinurhinus sumatranensis. I can't even say that right. So Rhinoceros unicornis and Dicerrhinus, Dicerrhinus sumatrensis. Oh my God, I finally got it out. That was disgusting. I should have practiced that a little bit before doing this question. Use information in table 1 to give the scientific name of the taxon labeled B in figure 4. So we go to figure 4 here and we use this information to tell us. Remember, we said there's no overlap, hierarchy, blah, blah, blah. Well, what we first of all need to identify, they've given us some clues here, guys. Why don't you pause the video, have a go? Remember, they've given us the binomial system. And we know in the binomial system, the one that's all in lowercase here at the back of these two bad boys, that is the species. So we know that because it's in lowercase. That is the species. The genus has a capital letter. So we have the species here. That is species there. That one there. That is the species. So what does this make rhinoceros or Dicerrhinus? What does it make those? Those must be the genus right here, peeps. That's the genus. And so what does it make this layer, this oval here? What does it make this? This must be the next one up as we go, family. And then what does it make this layer that is labeled B here? What's the next one up from family? It's the order. So that must be the order. So we're going with order because we just worked our way back from the binomial name, the species to the genus, species to the genus, to the family, to the order. And there we go. Lovely bubbly. Right. Let's go and type that in the answer spot. Bro. There we go. It is the order. And let's read the next question. Draw an oval on figure 4 to show the species rhinoceros. Oh my gosh. Rhinoceros sondacus. So rhinoceros sondacus. So we need to draw an oval to show that within this diagram. Let's do it, peeps. Let's do. All right. It tells us it's rhinoceros sondacus. They're testing you on the binomial. So rhinoceros is the genus, sondaceus or sondacus or sendaicus, however you want to say that, is the species. So we need it to be in the genus of the rhinoceros. So over here, and then we need to write sondacus in there. And I can't already, without looking at it, remember how that was spelled, but sondacus, something like that. Um, Something like that. I think my oval needed to be a little bit bigger. So let's just oval that bad boy around there. Oh my days. How do I do that? I get rid of that. And then I'm going to put this oval around here. And I'm going to lose a mark. Why am I going to lose a mark? Because it must not be overlapping or touching this oval. See there where I've touched that oval? That is not going to get me a mark. So just draw a separate oval and let's label it S and let's keep it like that because we don't want the examiner to punish us because we're showing that it overlaps. And the specific thing is it mustn't touch this oval. Okay, beautiful. Let's move on. Oh, my days. Okay, what question are we on now? 5.4. Scientists investigated phylogenetic relationships between individuals of 5 species of rhinoceros. The scientists determined the DNA base sequence of the site B gene of each rhinoceros, compared each CYTB DNA base sequence with that of one Indian rhinoceros called the reference rhinoceros, calculated the percentage difference between each site or CYTB DNA base sequence and that of the reference rhinoceros. So we've got Indian, Javan, Sumatran, white and black, the different species investigated. We've got the percentage difference in DNA base sequence compared with the reference Indian rhinoceros. So look, the Indian rhinoceros that they compared to the reference Indian rhinoceros has a 2% difference. So it's not exactly the same as the reference Indian one, even though same species. The Javan's 5% different, the Sumatran 13%, the white and the black are 14% different. What can you conclude about the likely phylogenetic relationships between these species? Evaluate your conclusion, 4 marks. So you're really getting like 2 to 3 marks for concluding, which is just saying what you see. That's the easy marks. Just say what you see in the data. And then the evaluation, like 1 to 2 marks evaluating, is saying what's wrong with the data or why might you be cautious about your conclusions. So straight away, let's get to the conclusions. Which one has the least difference between the DNA base sequences and the reference is going to be the Indian, but that's the same species. So we might say that's variation within a species. That's like intraspecies variation. The black and white have the most variation in their base sequence compared to the reference Indian rhinoceros, and they have a 14% difference. The Javan, 5% difference, so is the least of different species to the Indian. So we might think that that is more closely related as it has a more similar DNA base sequence. That might be our conclusion. And the white and the black, we might say, is the least evolutionarily, is the least closely related, evolutionary speaking, because the DNA base sequence has more variation, 14% variation. So those would be my conclusions. But then we would have to evaluate as well, right? I mean, the Indian had least difference, so we know that there's variation. Another conclusion is there's variation within the Indian rhinoceros species, and we know that because the Indian species was 2% different than the Indian reference rhinoceros. So we know that there is base sequence variation even within the Indian species. So these are all certified conclusions we can make, and I've done 3 there, but there's more that we could say just by looking at that data. Now, what aren't we told? We are told if we evaluate our conclusion. So it's kind of like good and bad, isn't it? When you evaluate, you're thinking about what's good, what's bad. Let's have a little look. Okay, we don't know the sample size, right? They haven't told us they did 5. They looked at the DNA base sequence of 5 rhino from each species and calculated the mean percentage difference. Looks like they only did 1, so we don't know the sample size. So that's a negative thing. We don't know how many rhinos they used to calculate the mean percentage difference. We don't even know if they calculated a mean percentage difference or if they just looked at 1 rhino from each species. Okay, so that's a weakness. What else is that's my main weakness with it. We don't really know the sample size. We also actually look at the white and black. We don't know the it appears like they are the same distance from the reference rhinoceros, but we don't know the DNA base sequence. We only know the percentage difference. All right, a scientist obtained a rhino horn confiscated from poachers and wanted to identify the species of rhino that was killed for its horn. He used the procedure described in 5.4 and calculated the difference in the CYTB DNA as 14%. What can you conclude from this result? Explain your answer. Suggest a change to the procedure that will more precisely identify the rhino. Right, look, it has to be black or white. They both have a 14% base sequence difference to the reference rhino, and so did this mysterious horn. So that one's pretty easy. When we look at the second one, suggest a change in the procedure. How could we make it specific and find out if it was the black or white? Well, we should look at the DNA, specific DNA base sequence of the black and the white, and see which DNA sequence is more similar to the horn, has the same DNA sequence as the horn. We only know they're 14% difference from the reference. We don't know which bases are different, which genes are different, which triplets are different. We don't know that. So you want to find out which DNA base sequences are different and then match that to the horn, and then we can find out if it's black or white. All right, guys, don't forget to pause the video at the start of each question. A student dissected a sheep's heart. He prepared a risk assessment on carrying a scalpel using a scalpel. Complete table 3 by giving 3 control methods the student must use to reduce the risk associated with carrying and using a scalpel. Well, like I say, pause and have a go at this, but when carrying a knife, pair of scissors, scalpel, you could hold it so the blade is towards you and not sticking out from you, or you could put it in a sheath, a protective container so that it doesn't cut anyone. When cutting the heart, you're best off pinning it out to the board so it doesn't move. Pin it to a wooden dissection board. And then I would say use the scalpel by cutting away from your body. Don't stroke the scalpel towards your chest and stomach. That could end very badly for you. So those are just 3 of good practices to do when using cutting tools. Okay. We're not asked to say that the risk is that you will get cut and bleed, because it doesn't say what is the risk. It says how to reduce the risk. And the risk is obviously getting cut. Right, 6.2. A scientist measured the changes in blood volume inside the left ventricle of a mammalian heart during 1 heartbeat. Figure 5 shows her results. Use figure 5 to calculate the total volume of blood pumped from the left ventricle in 1 minute. So what's the total amount of blood that leaves the ventricle in a minute? We've got 0.8 seconds here, which is 1 heartbeat, it tells us. So we're going to do a mass calculation here. Why don't you pause the video and have a go? Okay, so what we essentially want to do is say, right, this took 0.8 seconds, but they've asked us for how much blood left in a minute. So I want to find out how many heartbeats take place in a minute. I do 60 seconds divided by 0.8. This has a heart rate of 75 beats per minute. And each beat, we now know how many beats there are in 60 seconds. We need to know how much blood leaves the ventricle with every one of these 75 beats. So we just need to do 75 times the volume of blood that leaves the left ventricle. Well, let's check it out. What's the amount it starts at? Well, it seems to start at 120 when it's filling up, so 120 centimeter cubed. And then what does it drop down to when it's emptied and then it starts refilling? So it seems to drop down to 40. No, 38. I'm going with 38, but my eyesight might be a bit crusty. So, because each one of those squares is worth 2. So I'm going 120 minus 38 is 82. So 82 centimeter cubed. I'm going to times that by 75 because it pumps that out 75 times in 60 seconds. And I'm going to get 6,150 centimeter cubed as my total volume of blood that leaves the heart in 1 minute. And that will get you 2 marks. Okay, don't forget to pause the video. Also hit the like, guys. Scientists investigate the correlation between the patient's blood pressure, BP, and the risk of these patients developing cardiovascular disease. They group the blood pressure measurements into 4 health categories: low, ideal, pre-high, and high blood pressure. Figure 6 shows the scientists' results. Relative risk of cardiovascular disease is on the left. We're not given numbers for it, but then we're given the blood pressure is low, ideal, pre-high, and high. And we've got a range there. For instance, low is 10 to 12, ideal is 12 to 16. Etc. In a separate investigation, a doctor measured the effect of blood pressure medicines on treating a large number of patients with a mean blood pressure of 22. So they've got high blood pressure, all the patients. Treatment 1 contained 1 medicine. Treatment 2 contained 3 medicines. He measured the change in mean blood pressure after the treatment. Figure 7 shows his results. We've got the change in the blood pressure. So after they've taken 1 medicine, what's that? That's like a minus 1.8 kilopascals of pressure, so it improved their blood pressure by 1.8. Whereas treatment 2 improved their the 3 medicines, treatment 2 improved their blood pressure by 4. Okay, the actual question says, using information in figure 6 and 7, evaluate the effect of these treatments on reducing the risk of developing cardiovascular disease in patients with a mean blood pressure of 22. So if you have a mean blood pressure of 22, how's it going to help? Which treatment? Pause the video, have a go. Okay, so let's have a little look. Let's make some statements that we can conclude. we can conclude treatment 2 is more effective in lowering the blood pressure than treatment 1, right? Because it went down by 4 kilopascals. Treatment 1 only went down by like 1.8 kilopascals or 1.7 kilopascals. After treatment 2, they have gone from high blood pressure to pre-high blood pressure. So there is a higher risk of cardiovascular disease still than normal. So even after treatment 2, they're still higher at risk than a normal person. So it doesn't reduce you to normal risk. However, what I would say is treatment 2 is way better than treatment 1. That's a good conclusion, because treatment 1, you end up still being in the high blood pressure category because it only moves you from 22 to 20.3, something like that, kilopascals. So really, we can definitely conclude that treatment 2 is more effective than treatment 1 due to the difference in the lowering of blood pressure and the fact that treatment 2 with the 3 medicines shifts you into a new blood pressure category with a lower risk. On a good note to this experiment, a good evaluation of the methodology, they used a large sample size, so it's representative of the population. What is bad? They didn't do a stats test to tell me whether having a 20 or 22 mean blood pressure in kilopascals is a significant difference. Is that a significant difference between the means? We don't know, because they didn't do a stats test like a t-test or something to tell whether these means are significantly different. So yeah, yeah. We also don't know how long. They don't tell us how long this was done for or how long they were having the treatment. So, and we don't know any side effects. So if we want to make any conclusions about the effectiveness, it's not very effective if all the patients died 3 days later from liver failure or something. So we don't know about any side effects. So that's what I do for my answer there for 4 marks. And feel free to check out the mark scheme. There'll be some extra things you could have written. Okay, let's get into this question. Seboni. Figure 8 is a photograph of a vertical section through a leaf observed using an optical microscope. That's a light microscope. Figure 8, we can see here, it looks like the cross section of a leaf. Yep, we got all our different stuff. 7.1, in the box below, here it is, produce a scientific drawing. I'm going to try and read that again without stuttering. Produce a scientific drawing of the arrangement of tissues shown in figure 8. Label one of the tissues in your drawing. Do not draw individual cells. And that's worth 3 fat marks. We just got to label one tissue. Dead easy. All we got to do here, guys, is not draw the individual cells, but basically show the different sections. So working, sorry, flying in my head, working off of the individual, working off my GCSE artwork, we're going to bust this out. So that was a nice squiggle over there. Let's move this back up. I don't know what happened there. Flopping around. No, did he? Right, so here is my first section, this band here, and I'm going to label all of them. So that's my first section. We don't need to label individual cells. I'd call this the upper epidermis, and of course you've got the waxy cuticle on top, but this is known as the upper epidermis. You'll know this from GCSE. These are the colorless or transparent cells that let light through for photosynthesis. I've then got another band of these rectangular or columnar. How do you say that? Columnar? No, columnar. Oh my gosh, they look like columns, these cells here. Hopefully, oh gosh, I can't say that. Anyway, rectangles that are like standing up with the short side at top. Oh gosh, I can't even speak properly. Let's get over that. Right, this is the palisade tissue. Palisade tissue, and it contains, guess what? Palisade cells. We may want to call it palisade mesophyll because it's A level. So let's call it palisade mesophyll tissue. And these cells are the ones which have chloroplasts in them for absorbing light for photosynthesis, tightly packed together, not to allow any light to escape the clutches of their chlorophyll. And then we have this big layer, which is the spongy mesophyll. So I'm going to draw another box. Another long rectangle. These aren't so rectangular, these cells are like all over the place. You can see these air gaps. That's why we know this is the spongy mesophyll. So spongy mesophyll tissue. Again, do you need to oh my days, check that out with a double I. Oh, sometimes I despair with my drawing. Okay. Let's move that over there. Spongy mesophyll tissue. And then what have we got at the bottom? We got the opposite of that. So we need to put that stuff in at the bottom. The thing with the stomata, guard cells, we have the lower epidermal tissue or lower epidermis. So I think any of these four labels is going to do, folks. Now, I don't know if I'll lose a mark, but I think that looks like a vascular bundle there. Would I be allowed to just like bus a vascular bundle out there? I don't know whether I'd have to check the mark scheme for that. I'd probably just go for one of these layers of tissue. The vascular bundle appears to be inside the spongy mesophyll, so I'm going to put the vascular bundle, and that is made up of the phloem and the xylem. So phloem tissue and xylem tissue. So maybe we could label either of those. Might be a little bit complicated. Okay, I'm going to go with 3 marks. One for the 4 sections, roughly the same size. So thin, thin, fat, fat. I probably needed to make that fatter to guarantee myself the size mark. Let's do that again and just make this one roughly to scale. I know it doesn't have to be to scale, but the spongy mesophyll is a fatter layer than the palisade mesophyll. So I just want to make sure that this layer is fatter than this layer of cells here. Okay, cool. I think that's going to be good enough. That's going to, the right sizes, 4 sections, and any one of these labels. Let's give myself one for lower epidermis. There we go. 7.1. Done, done. All right, let's move down. What next? What next? A scientist investigated factors that affect the shelf life of cut flowers. A cut flower is part of the stem with a flower bud attached after it has been cut from a plant. The shelf life is the number of days the cut flowers are in good enough condition to be sold. He took 12 cut flowers from a rose plant, determined the mean number of stomata per millimeter squared on the leaves, determined the transpiration rate for each cutting, stored the cut flowers on a shelf in a brightly lit room, determined the shelf life of the cut flowers. Table 4 shows his results. Month cut flowers were obtained December or April. The mean number of stomata were 23 on average to 20, and we've got some standard deviation there. We've got a range around that. Mean transpiration rate, centimeter cube per day, plus and minus 2 standard deviations. So we've got 20. So there's a big difference here in the mean transpiration rate, even though not that much of a difference in the stomata. And then the mean shelf life, we can see that it appears to be a big difference in the mean of the shelf life. Okay. And we've got plus and minus 2 standard deviations from the mean. And this covered over 95% of the data before you all get upset and say, they didn't do a stats test. Well, this was done with 95% confidence limits. So let's go. Because you know it's going to be an evaluation, people. Oh, no, it's not an evaluation. I will shut up. Slap myself in the face. Using information in table 4, what can you conclude about the effect of different factors on the mean shelf life of cut flowers? Explain your conclusions. So we're going to need at least 2 conclusions with 2 explanations. I would say mins. So let's have a little look what we are going to talk about. So guys, I'm going to use this page with the stuff on to write my conclusions and explain them. So what I want to highlight first of all here, guys, is we've been given this data for a reason. So let's check out the mean number of stomata and make a conclusion about the mean number of stomata in December and April. So if I look there and you've got 23 as your mean, that goes up to 25 plus 2 deviations and down to 21. Now let's compare that to April. So the April mean is like 20 there, but it goes up to plus 3. So what have we got between these 2 means? We have overlap between the standard deviations, and that goes down to 17. So there's definitely overlap here. Look, that is overlap. So my first point would be there is no significant, oh my days, there is no significant difference in the mean number of stomata between April and December. Okay, and so this has no effect. This has, should I say no effect? This has no significant impact on shelf life, therefore, right? This has little effect on shelf life. I can't spell effect. Or I should maybe no significant effect on shelf life. So that's my conclusion. There's no difference in the means. Look, because there's overlap. Let's check out the next bit. I'm going to do this in green because we like multicolors. Mean transpiration rate, centimeter cubed a day. We've got 22. So I've got to write that over here. 22. We've got +3, so it goes up to 25. And we got -3, so 22 minus 3 is 19. Let's check out this bad boy, 17, and down here, 7. Gosh, what are you doing, Tom? 15. Okay, there's no overlap. Look, that goes up to 17 at the most and down to 13 at the least. There's no overlap between the mean transpiration rate in December and April. So there is a significant difference. So my point I'm going to make about this data, I'm going to also write in green. So there is a significant difference in the mean transpiration rate between April and DEC. Okay. I wouldn't normally write DEC, but I'm running out of room. So you won't get the marks if you start writing DEC, so don't write DEC. There's a significant difference in the mean transpiration rate between April and December. So I think as there's a significant difference, that has an effect on shelf life. This therefore impacts the shelf life. So it's the shelf life is probably something to do with this difference in mean transpiration rate. We proved there's a difference in mean transpiration rate. And if we check out the shelf life, we can see straight away there is a significant difference in shelf life in the months. Okay. So that is 5 with up to 6 and down to 4. And that is for the December. Then if we look at the April one is nowhere near it. The April one lasts 16 on average days, and it goes up to 18. The range around it is 16 to 14. So the minimum it lasts with 14 days, maximum this 6. We got no overlap. Okay, there's no overlap. So we can make a conclusion here that there is a sig diff in shelf life between April and December, i.e., I could say the ones in December have a significantly longer shelf life. The ones in April, sorry, have a significantly longer shelf life than the ones in December. So yeah, significant difference in shelf life between April and December. And I think this is down to the mean transpiration rate, to be honest with you. Yeah, whoa, the stems in April last significantly longer. And is it linked to, I don't know, and linked to shelf life? Is it linked to shelf life? Okay, that's what I would go with, folks. So yeah, there we go. They did it with 95% confidence limits, so all of these things are justified. Okay, let's move on. Right, where are we? Evaluated our conclusions. Last part. Other than a change in temperature, give one change a scientist could make to the environmental conditions to increase the cut flower's shelf life. Explain your answer. Well, let's just take a look here, guys. So the ones which had a low transpiration rate had a longer shelf life, right? So when the transpiration rate was significantly lower, which we've got there, it meant a longer shelf life. That should have been clearly one of my conclusions. Hopefully I made that. But here we go. So let's explain it. So why do we think? So we need to talk about the factors that could lower the transpiration rate and then explain that will increase the flower's shelf life. So I reckon less light would mean, now it says give one change. I'm going to give you more than one. So why don't you pause the video and write down all the changes? Okay, I'm going to give you all the changes. So you could go less light. That means less stomata open. lower rate of transpiration. Okay, longer shelf life. Stomata are triggered, they're triggered by the light. Another point I could make is we don't want still air or low airflow conditions. So don't leave them near the window, near a draft, as that will mean lower rate of transpiration. What's the explanation? Why do you get a lower rate of transpiration with low airflow conditions? Is because you have because low airflow means low or a shallow concentration gradient. Concentration gradient reduces water loss by evaporation, I don't know, diffusion, right? You get lower diffusion gradient of the water. Why is the water diffusing? Because it's a gas. It's water vapor in the spongy mesophyll. It's not leaving the plant by osmosis into the air. It's leaving by evaporation or diffusion. So that would be my reason. And so longer shelf life. Now, they didn't say to mention temp, did they? No, other than temp. So that's less light, low airflow conditions. What else? What else? What else? What are the four main factors? It's humidity. Oh yes, humid. Keep in a humid environment. Keep in humid environment. If you go down the humid environment again, this lowers the concentration gradient between the the spongy mesophyll or whatever. It lowers the concentration between the leaf and air, so less water or a lower rate of evaporation, so or diffusion, so lower rate of diffusion of water um, means less transpiration and longer shelf life. Oh, I've probably longed that out with loads of spelling mistakes and stuff, but never mind, peeps. So the idea is if you lower the concentration gradient, then less water is going to want to leave the leaf, right? And how does that work? Well, you've got your leaf over here, and let's put some water molecules inside that leaf. So here's our lovely water inside the leaf. If we have a steep concentration, water is going to diffuse rapidly out of the stomata down a steep concentration gradient. If we say, now let's keep it in a humid environment, like down here, then we're going to be like that. Why is the water going to want to leave when it's almost like that? We're going to reduce the concentration gradient and make it a lot less steep. The other thing we can do is have low airflow. Why does low airflow matter? If you keep your plant by the window, then the wind will come along and maintain a steep concentration gradient by removing the air outside the stomata and make a steep concentration gradient so more water will diffuse out of the stomata. So you want to minimize that concentration gradient. You want to make it very low between the leaf and the air, and that will extend the shelf life because low transpiration rates equals long shelf life, and that should get you all of the marks, all of the marks, all of the marks. Okay, what are we into now? We got question 8. So guppies are small fish with brightly coloured spots on their body. The number of spots on a guppy is controlled by genes. A scientist investigated guppy breeding in a large population of guppies in a fish tank. Here's my little pink guppy I'm drawing. The fish tank contained brightly coloured stones. Each generation of breeding, she counted the number of spots on every guppy. So essentially, she's put her little guppy fish inside a tank, put some coloured stones in there, and then every breeding season, more guppies arrive, and each time she counts the number of spots on the guppies, based on the fact that there are coloured stones of different brightly colours inside the fish tank. And so we've got a graph here, figure 9, with the mean number of spots per fish and generation on the x-axis, and we can see that over time, so our generation after generation after generation, the number of spots on the guppies increases. The scientists concluded that the mean number of spots on the guppies changed in this fish tank because the brightly coloured stones had affected the behaviour of the guppies. The guppies did not behave aggressively towards each other, so they weren't killing the ones without spots, and their feeding behaviour did not change. Suggests one type of guppy behaviour that could be affected by the presence of the brightly coloured stones. So this is a little bit complex. Pause the video, have a go. Okay, what we're looking at here is reproduction, mating behavior, or breeding behavior. So essentially, the guppies, they told us that it wasn't down to aggression, territory. They told us that it wasn't down to feeding, what they're eating. So what's happening is the ones with the brightly coloured spots are reproducing with other ones with brightly coloured spots. So it's a mating behavior that's changed. 8.2 suggests a further investigation the scientists could do to confirm that the brightly coloured stones had affected the guppy behavior. A null hypothesis for the investigation. You get 1 mark for suggesting an investigation to prove it was definitely the brightly coloured stones that caused them to increase in spots or change their behavior. And can you come up with a null hypothesis for the investigation? So why don't you pause the video and have a go? Okay, so for a further, how can we prove that it was the brightly coloured stones that affected the guppy behaviour? Well, essentially, they already carried out an investigation where they put brightly coloured stones inside the fish tank, right? And they put those guppies in there, and they proved that each generation the number of spots went up. How can we prove it was the stones? Well, we can get a second fish tank without any brightly coloured stones. We can put the guppies in there, and then we can see, does the number of spots on the guppies with each generation also increase? And this will prove if it's the stones that are affecting the behavior or if it's something else in the fish tank affecting that behavior. So your answer here, you want to say repeat the investigation with a fish tank or an aquarium which does not have any brightly coloured stones in, or the stones are all a dull colour, or they're all just a plain colour, no stones or just plain stones in it. And then check the with each generation, measure the mean number of spots on that generation of guppies, and do that for several generations to see if something else is affecting the behavior. And then the null hypothesis should be that the stones have no significant impact on the mean number of spots of guppies with each generation, or have no significant impact on guppy behavior. In fact, I need to change that because it's not just normal stones, is it? It's brightly coloured stones. So we need to say our null investigation is the presence of brightly coloured stones or coloured stones has no significant impact on guppy behaviour or on the mean spot number. I probably should say on the mean number of spots with each generation. And there we go. All right, let's get on to the last part to this question. 8.3. The scientists repeated the investigation of guppy breeding in an identical fish tank, but added predators of the guppies. That's a bit mean. Into the fish tank at the 4th generation of breeding. And so we can see along they go, their spots are increasing, number of spots on the fish is increasing. Then suddenly, once they've introduced the predators on the 4th generation, the guppies with the number of spots seems to go down with each generation. So name the type of selection the scientists investigated in this fish tank. Explain how this selection affected the frequency of alleles in the population of the guppies in the fish tank. So it appears like if you have spots, you're more likely to be eaten by the predator. This is directional selection, where it actively selects for fish without spots. So if you had spots, you're going to get caught by the predator and eaten. So that's not stabilizing selection, which would take you to the middle, having some spots. This is to the extreme. This favors fish with no spots at all. And so that is directional selection. It's when it goes to one extreme or the other. Furry, no fur. Spots, no spots. That's always directional when it forces natural selection in a particular direction to one of the extremes. So what's the explanation for this natural selection process? So if you know this from GCSE, if a guppy has the allele for colour spots, then it's more easily seen by predators. It's got less chance of surviving, less chance of breeding, less chance of passing its spotty allele on to its offspring. Another way to word this is that guppies that don't have the allele for coloured spots or have the allele for plain scales less easily seen by predators. Therefore, they have a greater chance to survive, more chance of reproducing and passing on their allele for no spots to the next generation. And so that's called natural selection as well. That might be a mark on there just saying by natural selection. Okay, let's slap their marks down and then get on with question 9. Getting tiring now. Last question, folks. Last question. Let's go. Right, scroll down. Question 9, 9.1. Describe how the structure of the insect gas exchange system provides cells with sufficient oxygen and also limits water loss. It's worth 5 marks. You kind of get 3 and 2 or 2 and 3. So, two things about the gas exchange system and how it can get oxygen quickly to meet its metabolic demands, and also how it can limit water loss. Water loss? Water loss. So here is my crusty little insect. Okay, yeah, doesn't really look like an insect. And what they have are little holes along the side. So we're getting into this. You might want to pause the video and have a go at this on scrap paper because these are some content questions. This is very good for your paper 1 A2 biology because the last 3 questions are 5-mark content ones. So the insect has spiracles, tiny holes on the side of the insect's body. So that's going to get you a mark that it has spiracles. Oxygen can just diffuse from the air into the spiracles down a concentration gradient all the way along to respiring cells. Now, how can it travel that distance? How can it travel that distance and meet metabolic demand, get there quickly enough? Well, the tracheoles and the trachea, which are the tiny little tubes that come off of the spiracles and go directly into the tissues and the cells of the insect, have a large surface area. So that is going to mean we get rapid diffusion of oxygen down a concentration gradient to respiring cells. Um, yeah, I haven't put concentration gradient. You should just put down a concentration gradient there. That would be important. And that the spiracles are tiny holes along the side of the body of the insect as well, because they might say, you know, the word spiracle, but you don't know it's a hole. Okay, the trachea and the tracheoles are the tubes. The tracheoles are the smaller tubes that branch off of the trachea, and they're filled with a little fluid and which allows the gas to dissolve. So they're moist, so they allow gases to dissolve more easily, and so they're more likely to get sufficient oxygen to meet their respiratory needs. Now, the tracheoles have thin walls, just like all exchange surfaces, whether we're talking about the alveoli, the squamous cells in the lungs, or fish gills, whatever exchange surface we're talking about. So we can say that the tracheoles or the tracheoles have thin walls, which means there is a short diffusion pathway, so oxygen can diffuse into the respiring cells of the insect rapidly. What else? What else? What else can we say? What else do you need? You need a good blood supply, a large capillary network. They don't have a good blood supply or large capillary network because they have an open circulatory system. So we don't want to put that. So you really want to stick to the short diffusion pathway with the thin walls or the large surface area due to the tracheoles and trachea spreading out. Now, to reduce water loss, they can actually close their spiracles and that will reduce water loss. But like stomata on plants can have hairy stomata, they can also have hair around the spiracles that will trap water and create a high humidity outside the spiracle, reducing the concentration gradient from inside to outside of water. So less water will diffuse out. So those are some other reasons. Okay, let's move on. Now, looking at 9.2, describe how humans breathe in and out. So why don't we pause the video, grab our pen, grab our paper and have a go at this one. It's a 5-marker. Again, it's great practice for your A-level biology paper 1 right at the end. Lots of content questions, 15 marks of content questions, normally 3 5-markers, sometimes a 6, a 4 and a 5. Who knows? Right, breathe in. Let's deal with the breathing in. Now, check what I've circled, in and out. You've got to cover both aspects here. If you only cover one aspect, you're not going to get all 5 marks. The maximum you'll get is 3. So breathing in, we're looking at the external intercostal muscles contract. What else contracts? Well, the external intercostal muscles are the ones between your ribs. Now, when they contract, the ribs move up and outwards, and that's going to increase the volume in the thoracic or thoracic cavity. And then the diaphragm contracts and flattens. That's the muscle at the bottom of the thorax. That flattens. And what that does is that increases the volume inside the thoracic or thoracic cavity. And this decreases the pressure. So it makes the pressure less because a bigger volume, those air particles that are inside the thoracic cavity have a bigger space, less collisions with the walls, etc. You get the idea. So this reduces the pressure, and the pressure actually falls below the atmosphere relative to the atmospheric pressure of 1 atmosphere or whatever it is, 100,000 pascals. The pressure inside the lungs falls below that, and therefore air moves in down a pressure gradient into the lungs from a high atmospheric pressure to a low pressure in the lungs. So the whole physical process of making your ribs go up, diaphragm flatten, is to increase the volume of the thoracic cavity, reduce the pressure, and that will force air in. It will like rush in like a wind from high to low. Okay, so that's covering the breathing in, and we could probably get a maximum of 3 marks for one of these, as it's 5 marks. Maybe they'll be generous and do 4-1, but let's have a little look. We're going to do breathing out, so we can just say all the opposite things. So it's the internal intercostal muscles. Sorry, the external intercostal muscles relax now. We don't want to talk about the internals. We do use the internal intercostal muscles for forced expiration, but we don't need to talk about that here because they're not asking us about forced. So external intercostal muscles relax, the ribs move down and inwards. The diaphragm relaxes and it domes. So this is a bit confusing to people, but the diaphragm actually kind of moves up and becomes like a dome when it relaxes. And this is going to reduce the volume in the thorax or thoracic cavity, and that will increase the pressure because there's more collisions with the air particles in the thorax and the walls of the cavity. And so, therefore, the pressure increases inside the thoracic cavity and it's greater than the atmospheric pressure. So, the atmospheric pressure is 100,000 pascals or 1 atmosphere. So, you can imagine the pressure inside the thorax maybe goes up to 110,000 pascals or something. So, now air moves out down a concentration gradient. And that, my friends, is the end of the paper. Don't forget sharing, caring, hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.